Hello everyone, this is part 2 of resting membrane potential. Those who haven't watched part 1, I'll provide the link in the description. You guys can see from there. In the previous video, we talked about sodium potassium pump and its contribution to RMP. We saw that directly this pump does not help much to create RMP, that is, it produces electronegativity of only minus 5 millivolt. See, the sodium potassium ATPase gene was our first gene in discussion, and which, when expressed, it resulted into sodium potassium ATPase, which helps in concentrating K plus inside the cell and sodium outside the cell. Now, let's talk about these two genes which I have drawn over here. And let's suppose this gene over here, it also expresses and it makes proteins. Those proteins, they also get stuck into the plasma membrane, that is, they get embedded into the plasma membrane. Let me show you this protein. Basically, this protein is made up of peptide chains. One, two, three, let's say, whatever their number is. What I mean to say is, this gene is making some peptides, and those peptides, they clump together and they make a pore, and this pore is embedded into the cell membrane. Basically, these pores, they are lined by electronegativity from inside. Due to that, it will attract cations. These channels or pores, they allow the movement of K plus across them, but they do not allow Na plus to move across them. So if we want Na plus to move across, we would have to make special sodium channels using a different gene, that is using this gene. This gene, it will make proteins that will get planted into the membrane and which will allow movement of sodium. But these channels, they do not allow the movement of sodium. They allow only K plus to pass across them. But in this hypothetical cell which we are studying, let's suppose sodium channel making gene, this one, is not working. Only K plus channel making gene is working. So now what is happening? Only this gene is expressing and it is making proteins, this one, and they are basically K plus channels. In this cell which we are studying right now, sodium channel making gene is not working. All right. Now see, sodium is trapped outside. It cannot go inside using these channels, and if somehow sodium gets inside, it will be again thrown out by this sodium potassium ATPase. But on the other hand, if you see, potassium is concentrated inside, and not only it is concentrated inside by sodium potassium ATPase, it is also allowed to move out, or we can say it is allowed to move across the membrane in either direction, because this pump, it does not determine in which direction K plus will move, it just allow K plus to move across them. All right. So K plus, it can move inside the cell also and it can come outside the cell also. It depends upon K plus in which direction it wants to move. We'll see what are the factors that determine the movement of K plus later. See what happened up to now. Sodium potassium ATPases are planted into the membrane, and due to that, cell started throwing the sodium out and concentrating the potassium in. Now, sodium is concentrated outwards, and this sodium, it would love to go in along its concentration gradient. Things, they move along their concentration gradient, that is from their high concentration to their low concentration. So sodium is trying to come in, and K plus that has been concentrated inside, it would love to go out along its concentration gradient. But the problem is, this cell which we are studying right now does not have sodium channels, because this gene which is responsible for making those channels is not working here. All right. Now, these special type of channels which you are talking, they are known as potassium leaky channels. They are basically ungated or unregulated channels, that is, they are open all the time. And also one more thing, the movement of K plus across them is passive. Okay, this is passive, not active. All right. See, next we have is, there are many type of channels a cell can have. I'll discuss them later, but I'm giving you a brief overview here. So the first one let's talk about is the ungated channels. They are called ungated channels because they are unregulated, and these channels, they are working all the time regardless of the situation. So you simply call them leaky channels. All right, next we have is there are some channels which have special gates. These channels, they have special gates, and these gates only open at a particular voltage. So these channels we call them as VGC, which stands for voltage gated channels. Then there are another group of channels, and these channels, they have a special arrangement where a particular substance bind. See, when this particular substance bind, then only it opens or closes. Such channels, they are called as ligand gated channels. Ligand means a specific substance which can bind to this and change its activity. Now, see, cell membrane, it can have many type of channels, but broadly they can be categorized into three categories, that is ungated channels, VGC, voltage gated channels, and ligand gated channels. All right. See, ungated channels, that is open all the time. Then there is VGC, they are only operative at a specific voltage. And then there are these channels, they are only operative when a specific substance it binds to them. See, this specific substance, it can be a neurotransmitter, or it may be, this ligand gated channels, it can be activated by a hormone also, or, some secondary messenger can also activate them. Now, let's get back to this potassium. We'll see the behavior of this potassium. See, what is happening is there is a lot of potassium inside the cell, and the cell membrane is now permeable to potassium because of the K plus leaky channels. There is also a lot of sodium outside, but the cell membrane is not permeable to sodium. Now we'll see what really happens to this cell and how this K plus behaves. See, this potassium is willing to go out. It is ready to rush out because concentration gradient is a force which is taking it out. All right, and it is because the concentration of K plus is more into the cell and it is less outside the cell due to activity of this pump, that is sodium potassium ATPase. So of course, potassium will love to move from its high concentration to its low concentration if there is a pathway available, and luckily this cell, it has a lot of K plus channels, so K plus has an option to leak out. Now, what is the driving force for this potassium to leak out? That is its concentration gradient. I'll make a big channel here so you all can see what really happens. Now, this K plus has a very strong concentration gradient outwards. But remember, this pump, that is sodium potassium ATPase, due to this pump, the cell interior is already electronegative by minus 5 millivolt. All right. Now, potassium is a cation, right? It is positively charged. So when these positive charged ions, they are trying to move out because of their concentration gradient, this electronegativity will try to hold it in. But concentration gradient is trying to take the K plus out. So now what happens is this K plus has a very strong tendency to go out due to its concentration gradient. But there is also a small force which is acting inwards due to this minus 5 millivolt, which is called EG, that is electrical gradient. See, this concentration gradient which is acting outward, it is very strong, but this electrical gradient which is acting inwards is very weak. So net electrochemical force which is driving this K plus is towards outside. Because this concentration gradient is very strong, so what will happen is this K plus ion, it will start leaking outwards. See, as more and more potassium is leaking out, is going out through these potassium leaky channels, what is happening is interior of cell is becoming more and more electronegative. Let's just say from minus 5 millivolt to minus 10, minus 25, all right, minus 50. So basically, more the potassium diffuses out, more the interior of the cell becomes negative. Now, one more thing, this potassium inside the cell, this is basically not alone. This is actually, this potassium inside the cell, this is actually bound to anions. Alright, I am representing anions by A minus. This is your non-specific anion. Anion means what? It is a negatively charged ion, right? So this anion, it consists of two things. That is phosphates and proteins. See, phosphates, they are negatively charged, and these proteins, they are made up of amino acids. tons of amino acids actually, and amino acids, they have a lot of negative charges on them. So basically both are negatively charged, both are anions, and they would love to interact with this potassium, that is a cation. So whenever this K plus leaks or diffuses out, you must be thinking that these anions associated with it are also going to leave. But no, that's not the case. These anions, they cannot leave the cell because they are too charged and they are too big to leave the cell. Alright, so whenever K plus leave the cell, whenever this K plus leave the cell, they always leave behind an unoccupied, unbound anion. Now, every time this potassium it leaks outside the cell, what is happening is it leaves behind these unoccupied anions, and due to that, inside of the cell is becoming more and more electronegative. All right. See, cell was at -5 millivolt, then potassium diffuses out -10, more potassium diffuses out -25, then more potassium diffuses out -50. So what exactly is happening is K plus, and this K plus is diffusing out, and potential in the membrane, it is changing. Basically, this potential is changing due to diffusion of this K plus. See, it is very easy to understand that the K plus concentration across the membrane was different and the membrane was permeable to K plus. So K plus was moving out, and when it was moving out, then you can say potential in the membrane was changing. Now see this electrical potential which is building up or which is developing due to diffusion of this K plus, this potential it is called as diffusion potential. So as more and more potassium it diffuses out, cell will develop more and more diffusion potential due to potassium. So diffusion potential is developing. This is basically electronegativity which is developing into the cell due to diffusion of potassium outward. As the cell it is losing K plus outward, it is losing the cation outwards, and it is retaining the anions, because as I told you earlier, these anions they have a lot of charge on them and they are too big to leave the cell. So due to loss of these cations and retention of these anions, the cell is becoming more and more electronegative. Now see this concentration gradient of potassium, it does not change much because the total K plus present inside the cell is too much. But still the cell is becoming more and more electronegative due to loss of these cations, that is K plus. Now this small force which was acting inwards, which we discussed earlier, that is the electrical gradient, this will progressively get stronger, this will progressively grow because cell is becoming more and more electronegative due to loss of these cations. See, as cell is becoming more and more electronegative, this electrical gradient is progressively growing. It means as K plus is moving out, for remaining K plus to leave the cell, it is becoming really difficult. Like initial K plus would easily leave the cell because electrical gradient is not that much stronger. It is just due to -5. But as with time, a cell is becoming more and more electronegative, this EG is growing. Alright, so for later K plus to leave the cell, it will be really difficult because now it has to face a stronger electrical gradient. Again, see what happened. Let me repeat the entire thing. See, first of all, sodium potassium ATPases, they throw all the sodium out. So outside is the sodium rich area. Alright, and it concentrated the potassium inside. So inside is the potassium rich area. See, along the concentration gradient, this sodium, it would love to come in because it has a higher concentration outside and it has a lower concentration inside. So it wants to move from outside to inside. But what is happening is this cell which we are studying, the membrane, the cell membrane of this cell is not permeable for sodium because there are no channels available. Why there are no channels available? Because this gene which was responsible for making channels for sodium is not working. Now see this potassium which is concentrated inside, it would love to go out along its concentration gradient, and luckily this cell has potassium leaky channels and they are present in the cell membrane for the movement of potassium. Then we saw that this concentration gradient for potassium is outwards, and luckily the membrane is permeable for K plus, so K plus it starts diffusing outward. Now see, as potassium it is diffusing outward along the concentration gradient, interior of the cell it is becoming more and more electronegative. negative. So due to diffusion out of the potassium, the membrane is developing an electrical potential, and this electrical potential which is developing due to diffusion of potassium is called as potassium diffusion potential. All right. Now next is, let's just say concentration gradient in the beginning it was 4 and electrical gradient it was 1 due to minus 5 millivolt. Then as time passes and this time is very small. Also remember that there is a very little shift in potassium that occurs to produce a significant diffusion potential. Now, due to diffusion of this potassium outside through these potassium leaky channels, due to diffusion of these cation outwards, cell is becoming more and more electronegative. So let's just say concentration gradient is still 4, but electrical gradient it has now become 3. See, in the beginning concentration gradient was 4, now also it is 4. It is because I've told you earlier also, concentration gradient does not change much because total K plus present inside the cell is too much. And also there is very little shift of potassium to produce a diffusion potential. Also, what is the difference between diffusion of this potassium and this potassium? See, this potassium in the beginning, it slipped out very rapidly. But if you look at this potassium, then it slipped out but relatively slowly. Why so? Because concentration gradient in the beginning, it was 4 and it was balanced by electrical gradient which had a magnitude of 1. Then later on, concentration gradient is still 4 but electrical gradient, it progressively increased to 3. Electrical gradient is basically nothing but the resisting force. See, this concentration gradient, it wants to take this K plus out. But this electrical gradient is the resisting force and it does not want K plus to leave the cell. That's it for this video. We'll continue the rest in part 3. Thank you so much and do subscribe and like the channel.